Assalamualaikum and good evening to Dr. Andrew Nadine and my fellow friends. How Gregor Johann Mendel discovered his theory and benefited scientists. Gregor Mendel, through his work on paired plant, discovered the fundamental law of inheritance. He deduced that genes come in pair and are inherited as distinct units, one from each parent. Mendel tracked the segregation of parental genes and their appearance in the offspring as dominant or recessive traits. Among track experimentation in his monastery garden, Mendel discovered the universal concept of heredity. His research found that the inheritance of such characteristics in pair plants follow unique trends, thereby becoming the basis of modern genetics and contributing to the study of heredity. How the Mendel theory is applied to meiosis and how the meiosis process takes place. Gregor Mendel was a 19th century Moravian monk who demonstrated that the inheritance of traits. First, law of segregation. Each hereditary characteristic is controlled by two alleles which separates into different gametes. Law of independent assortment. The separation of alleles for one gene is independent to allele separation for another gene. C. Principles of dominance. In pairs of alleles that are different, one allele will mask the effect of the other allele. These laws are now known to be due to key events that occur during meiotic division. The law of segregation describes how homologous chromosomes are separated in meiosis. First, the law of independent assortment describes how homologous pairs align randomly during metaphase. First, third, the elucidation of the process of meiosis, we now know that there are certain exceptions to Mendel law. Genes that are of the same chromosome will not undergo independent assortment. Not all genes display a dominant hierarchy. Certain traits may display codominance or incomplete dominance. We can see the segregation, independent assortment, and principle of dominance from meiosis. Why chromosomes are important in mitosis? During cell division, it is essential that DNA remain intact and evenly distributed among cells. Chromosomes are a key part of the process that ensures DNA is accurately copied and distributed in the vast majority of cell division. Chromosomes in eukaryotes perform a useful function during mitosis, the process in which cells replicate their genetic material and then divide into two new cells, also called daughter cells. When the cell divides, the sister chromatic from each duplicate chromosome separate. Okay, explain the crossover during meiosis and its effect on organism. Crossing over is simple the exchange of genetic material between two homologous chromosomes to give rise to recombination chromosome. In preface 1, homologous chromosomes align lengthwise or pair with others, and exchange of genetic material between the two chromosomes takes place, which is known as crossing over. The pairing of the homologous chromosome is known as synapsis, and the point at which this chromosome pair with each other is known as a chiasma. The process of crossing over or recombination is initiated by the protein SPO11. This protein from a complete with other protein like RAD50 and MRE11A, and NBS1 is required for breaking of the double-stranded DNA. Certain isonucleases are required to bring about the deletion of the 5' in order to generate single-stranded tree tail. MRE11A has also been seen to pose certain isonuclease as well as endonuclease activity. The DNA recombinase like DMC1 and RAD51 then take over. These two proteins bind with a couple of other proteins and are required for the invasion of the non-sister chromatic. The MC1 is required to find the allele sequence on the non-sister chromatic. Already FISH1 help to bring about strand invasion of the non-sister chromatic in an ATP-dependent manner and well and in the search for allelic sequence. Next, the tree end of the invading strand is used as a primer for the synthesis of the complementary DNA on the non-sister chromatic that has been invaded, annealing the invading strand to it. As the synthesis of the complementary DNA sequence continues, it displays the original complementary strand. The displaced complementary DNA strand then anneals itself to the other that was originally complementary to the invading strand. The structure that is thus formed is known as a holiday junction. These interlocked strands are then nicked and ligated with to help of certain endonuclease and ligase. It must be noted that the creation of the single strand tree still take place in the non-coding part of the DNA or in the junced DNA. Crossover increase genetic variation in gametes and offspring. Crossover allows the chromosome you pass to your children to have some change from your mother and some from your father.